Good afternoon, and welcome to the National Bleeding Disorders Foundation Wednesday webinar. My name is Fiona Robinson, and I am the series host. In today's webinar, we have united a panel of hemophilia experts to tell us all about advancing my life, our future. This webinar is being recorded and will be available to the community on the NBDF website, bleeding.org, shortly after the webinar. We encourage you to ask any questions you may have today. You can ask your question by using the Q&A feature at the bottom of your screen at any time. We'll monitor these questions, and following our speakers' presentations, we'll put your questions to them. We have a wonderful panel here today, and I will just introduce Dr. Michael Recht, who's our Chief Medical and Scientific Officer of the National Bleeding Disorders Foundation, and I will hand things over to him now to get us started.
Thank you so much, Fiona, and welcome everybody to today's Wednesday webinar. As Fiona said, my name is Michael Recht, and I am the Chief Medical and Scientific Officer of the National Bleeding Disorders Foundation. We have a very exciting talk today. We are going to be talking about My Life, Our Future. Joining me on this incredible panel are some of the most important leaders in our field. I'd like to I'm not going to go through everybody's CV because I would be taking the entire hour to just introduce people. So I'm just going to introduce people by name, and then we're going to jump into the webinar. So joining us today is Len Valentino, Shelley Horowitz, Glenn Pierce, Mark Skinner, Jill Johnson, and Barb Conkle. The reason these people are all on the call is they have been involved with My Life, Our Future from its inception, and we're going to be talking about the origin story of My Life, Our Future and where we're hoping the project is going moving forward. What I can say in my career as both a treating hematologist and a clinical researcher, My Life, Our Future has probably been one of the most impactful programs I have ever been involved with. So I'm really looking forward to today's webinar. So I am going to pass it off to our panel. The one thing I would like to remind the attendees in today's webinar is we cannot answer individual healthcare questions. We cannot give advice to individuals. But any questions that can be addressed more on a population basis, we are happy to discuss. So thanks for joining us, everybody, and let's get off to the races.
Thanks, Mike. My name is Glenn Pierce, and I'm here to talk about the beginnings of My Life, Our Future. And so I'll start with where have we been over the past 20 years or so. And with the next slide. Next slide. Where did we need to go? No, oh, sorry. All right, we have to get our signal straight. So where did we need to go when we started thinking about this 25 years ago? Now, the next slide, please. I think that everybody is pretty much up to speed on what hemophilia A and B are, but just to make sure we're all on the same page, you know that hemophilia A is due to a deficiency of factor VIII, hemophilia B, a deficiency of factor IX, and together they comprise the two most prevalent types of hemophilia. Hemophilia A is quite a bit more prevalent than hemophilia B, and the prevalence figures are listed here. They're revised figures based upon a really seminal article by Iorio, Stonebreaker, and colleagues published in 2019 that really give a good definition of the global incidence and prevalence at birth of hemophilia. It's also important to recognize that we have a lot of variants in hemophilia, a lot of causes for hemophilia that I'll get into in a moment. And part of that is because we have a very high rate of change in the hemophilia genes, factor VIII and factor IX, as new humans are created. And so that rate of change continues to develop additional people with hemophilia that have no family histories. All of these changes are called DNA variants, otherwise known in the past as mutations, and there are many thousands of them that my colleagues will talk about. Next slide, please. So let's get some definitions straight here, so that you can understand, hopefully, the rest of the talk and the other speakers. We're talking about genotypes and genes. And what I want to first call your attention to is on the right-hand side. This is the code for life. And so we start with DNA, which is present in all of the cells in our body. And this DNA encodes 20,000 different genes that make us who we are. And it does that by taking the DNA itself, the genes themselves, and turning them into messenger RNA that then allows us to encode proteins. So if we have a variant in the factor VIII or factor IX DNA, that gets transmitted to the RNA, that gets transmitted to the protein, and then we have a dysfunctional protein, or a protein that either doesn't work or isn't even able to be made. So that's the basis for it. The genotype really reflects what the variant is in each individual patient, and it may be different in many patients. And so it causes hemophilia. A DNA variant is that change in the nucleus, in the nucleus, in the DNA, where there's lots of reasons for that we don't need to get into in any detail at this point, but suffice to say that many different reasons for the hemophilia phenotype or the clinical manifestations of hemophilia have been identified at this point in time, many thanks to the My Life, Our Future program. Next slide, please. So let's go through the who, what, when, where, why. And if we start with the why, the hemophilia genes were cloned in 1982 and 1984, respectively. And using pretty crude technology and crude techniques, they began to identify a series of mutations in some patients. Those were the variants that we now refer to today. But technology improvements by the end of the 1990s enabled people to detect these variants more easily. Part of that was due to the fact that the first human genomes were cloned in 2000 and sequenced in 2001. And so that gave us a lot of new technology in which we could take individual genes such as factor VIII or factor IX and begin to understand what causes the clinical manifestations in people with hemophilia. So understanding that relationship between the genotype and the clinical phenotype became important, and it became important for management of patients as well. This is now called precision medicine. You'll hear a little more about that. But we were really early in the process. We treated patients with precision medicine, as it was known, as it became later known, by simply looking at dosing factor concentrates to patients who perhaps had shorter PKs or longer PKs and tailored the dosing to them. So part of that is precision medicine, and it really allows us to understand what's happening with individual patients. Now, we were able to think about studying the hemophilia genes in the whole community because of the community itself. This is a really unique community where you've got this interplay between patients, patient advocates, the chapters of the NBDF, and the hemophilia treatment centers. And all three of those groups work in conjunction to further the goals of the entire community and allow us to think for the first time at that point about doing countrywide programs, research programs. So this can help individual participants in a program, individuals with hemophilia, but it also can contribute to the greater research questions for the entire population. Next slide, please. So this all sets the stage for the My Life, Our Future program. The groundwork was set in the 1990s as I mentioned. And we even had a MASAC resolution in 1998, which advocated for the value of genotyping in helping to better manage patients with hemophilia. We also knew at that time, in 1999, that clinical trials for gene therapy for hemophilia were just beginning. And while we've come a long way since then, it has proved to be important to be able to understand what the patient's genotype is as one considers them for gene therapy. And then we worked with Congress to get more support for national initiatives. And back in 2012, when it looked like we could develop a nationwide genotyping effort, we ran a survey through the NBDF, the NHF at the time, and the vast majority of community members expressed an interest in participating, in understanding what their genotype was. And the vast majority of hemophilia treatment centers also wanted to have that understanding and facilitate getting the data for the patients. The next slide, please. So what did the MLOF aspire to do? Here's the what behind the questions. Well, to really collect a large number of patients with hemophilia A, hemophilia B throughout the country. And really be able to put it all together to start to look for trends, look for patterns in the data, look for unique genotypes that patients had, and how that began to relate to their clinical progression for hemophilia. And so we also wanted to be able to look for modifying genes. So two people with an identical genotype, one person bleeds a lot, one person doesn't. What's the difference there? And how can we begin to address that difference for the benefit of the patient? And so as we think about gene therapy, there's a whole next generation of gene therapies that's coming along in preclinical development, starting to enter the clinic now, that are going to be looking at specific types of gene mutations and doing gene therapy on those specific types. So that's something that will be continuing to develop over the next 5 to 10 years. And of course, you can't do that unless you know what the genotype is. And then the last great thing about the program is that we began enrolling females who were at risk, or females with hemophilia, to begin to understand their genotypes and how those related to their phenotypes as well. And so that also became a core component of the program. With that, I'm going to turn it over to Mark Skinner to continue the discussion about the history.
So, good day to all. We have to recognize that the concept of my life, our future didn't occur in a vacuum, that it's part of a greater revolution in healthcare and science. And the notion of what once was called personalized medicine, now named precision medicine, really involves genomics, the kind of genetic data that my life, our future would be delivering. And by having this genetic data, the hope, as Glenn has laid out, was to really explore and unlock some of the mysteries of hemophilia using the anonymous data that was collected from quite literally thousands of people living with hemophilia. The build, please. Next, click. So my life, our future really reflects what's on this slide, that precision medicine is defined by the NIH as an emerging approach for the treatment of disease and prevention, taking into account the individual variables, the environment and the lifestyle of each person. So the goal of, or a goal of my life, our future, was really to allow us to deliver optimal care to the individual, to move beyond treating the disease of hemophilia generically, but to treating the individual uniquely based on their genes, their environment, their lifestyle. And so some of the kinds of questions which you might be pondering that we contemplated at the time are, why do some people develop inhibitors and others don't? Is there a genetic reason for this? Why do people with the same genotype have different factor levels? Why do some women, carriers of hemophilia, have low factor levels and others don't. And the number of questions that you can think about really could go on. The next slide, please. So it really is this, this nature of addressing the individual, going deeper into individual lives and allowing this research to support me as an individual living with hemophilia, but also to collectively contribute towards our future, to answer some of these bigger questions, hence the name, My Life, Our Future. Next slide, please. So Glenn mentioned that, in the beginning, there were 4 organizations that came together. We had a history of interest in the area, but really through the leadership of the names and the individuals that you see on this screen, this program really came to fruition. Biogen was an integral partner in stepping up and committing to fund the initiative, provided scientific collaboration and certainly financial support. Puget Sound really was the primary place for doing the genetic testing and the analysis and managing the research repository. The National Hemophilia Foundation, as it was named at the time, was responsible for the communication, the community engagement, and helping to recruit patients. And then, of course, Athen, as our data repository, was responsible for maintaining and collecting the data into a warehouse. Each of these organizations, or most of them, I think all of them almost, except Athen, have been renamed and have had new iterations. But it's important to recognize our founding partners and really the core of the groups that stepped forward to bring this. And of course, Barb and Glenn are both on the webinar today. Next slide, please. So this became quickly a countrywide, a community-wide effort. Our community, as has often been the case, have really been pioneers in the space of rare disease. And so our ability to do large-scale, data collection of phenotypic clinical data as well as genetic data is somewhat unprecedented. Others have done it, but to do it to the extent and scale that we did in the community really was a significant achievement. The ability and the of chapters and hemophilia treatment centers and researchers working together to advance our knowledge and the science behind the treatment of hemophilia is one of those things that we want to continue to try to achieve and build on. And it was designed to provide a response or a service to the individual, my life, as well as a service to science and research, our future. And so together, these twin goals, I think, really solidified the collaboration that we were looking for. Next slide, please. And before I hand it over to Barb, I really like this slide that, if we're not ready for research, and people that are interested in doing research are going to go somewhere else. So the notion of having a research-ready community, we have a rich data set, we have community members that are willing to be engaged in research. And so collectively, we had positioned the community such that we can use this as a catalyst to capture the advances in science as they come forward. And so I summarize this sort of in the way that if we have better data, if we have the better genomic data, then we can do better science. And then the better science will ultimately lead to better outcomes for those of us that live with a bleeding disorder. So I'm going to pass this on to Barb now.
Hi, I'm Barbara Conkle. It's great to join. And I'm just going to give a review or reminder of the MLOF program structure. So almost all HCCs across the country participated, and there was great involvement and collaboration with then NHF chapters. And then participants were approached. They authorized the testing. The Hemophilia Treatment Center drew the sample and sent it to what was then Puget Sound Blood Center and now BloodWorks Northwest. We did clinical genotyping testing and reported a clinical result to the center. And then individuals, parents of patients or people affected by hemophilia or potentially affected by hemophilia could give informed consent to participate in the research component. And with that, there were additional data collected into the Athen data set. and also the samples were stored. All of this from the our standpoint was people were given an MLOF number. So the identity of individuals was at the HTC and not at the program, and that continues. And then the research repository, which you will hear more about, was formed. Next slide. So MLOF was open for enrollment from 2013 to 2017 with completion of clinical reporting in 2018. There were 107 HCCs and over 11,000 participants received clinical genotyping results. About 2/3 were male, 1/3 female. About 80% tested for hemophilia A, which goes along with the prevalence of that versus hemophilia B. And we were able to determine a genotype in people with hemophilia, so with factor level less than 40%, almost all of the time. And about 80%, or over 9,000 individuals or their parents consented to research, which just goes to show the involvement of the community and the interest in research. And so now it's my pleasure to turn it over to Shelley Horowitz, who will speak about how MLOF affected her.
Thank you so much, and it's a pleasure to be here. I'm Shelley Horowitz. Hemophilia A is in 6 traceable generations of my family spanning 176 years. And I'm involved in many aspects of community, but today I'm here to share my personal experiences with My Life, Our Future. Absence of explanation is not explanation of an absence. And absence of an explanation for my bleeding was not an explanation of an absence of bleeding. When a situation doesn't make sense, sometimes it is unfortunately minimized. So in my case, my levels are lower, but not super low. They range from 11 to 22%. And when I verbally reported bleeds, I was told I could not be bleeding because it didn't make sense. And I was told if my factor wasn't working, it wasn't a bleed. I knew I was bleeding. And can you push forward on the slide? I've included some bleed photos. The 2 elbow photos in the center are actually right after it was hit on the left, and then 45 minutes later before factor was infused, which is the right elbow photo in the center. And I was on prophylaxis actually at the time those were taken. I was lucky and fortunate enough to speak with Jill Johnson at a conference, and she had heard me express my struggles and to be believed and get proper care, and asked if I had participated in MLOF. And she encouraged me to find my Life, Our Future report and told me that relevant variant information was included in these reports whenever possible. So right after that conference, I went home and I pulled it out of a file, my Life, Our Future report, and I immediately found explanations. I found qualitative issues, and I found references that led me to many peer-reviewed medical journal articles referencing the implications of the variant I have. And the variant I have is well known for causing reduced binding between factor VIII and von Willebrand factor. So at that point, once we had that information, pharmacokinetic studies were then run, which had been denied to me prior, and my half-life was not the expected 12 hours, but was 4.6. The My Life, Our Future report greatly accelerated the process of understanding and moving forward for me, and that combined with pictorial evidence and PK studies then expanded the picture. So in this case, and in my case, I needed, and my treatment team needed, to understand the implications of my variant in order to recognize I was bleeding and guide decisions about an appropriate treatment plan. And for me, this was absolutely life-changing. My annual bleed rate has gone down, and I'm actually in less pain today than I was when I was in elementary school. So for me, and for many, the gift that My Life, Our Future gave me was connecting me with information about my variant. And that gift created validation of my bleeding experiences that I was having, and it allowed for the possibility of change. It gave me access to relevant research, to hope. I was believed, and it opened the door to access care and to reduce bleeding incidents. Additionally, knowing our family variant also allowed for targeted testing when my niece was born, which was very important to our family. I am incredibly grateful to the team who made my personal participation in My Life, Our Future possible, and for the value that it continues to bring to our entire community. And with that, I will turn it to Jill Johnson.
Thank you. I'm always so moved, so I need a second. I'm going to speak a little bit about what the program found when we analyzed the data from the participants. It's many of you, Shelley, and many others. So this program absolutely cannot happen without the community coming together and participate. And we are all grateful for that community effort. And I think it's a good example of how we all should be doing service programs and research. So I would like to point out that the the analysis of the genetic findings of the entire program was published in 2022. We made sure this is open access, so anyone should be able to find this, and the references here. And I'm going to highlight a few key things that we found when we did this analysis. Next slide. So one was that we found a huge number of different DNA variants, different DNA changes. And of those, over 1,900 of them were ones that we thought were relevant to the person's hemophilia. So not every DNA change is important, but over 1,900 of them are ones that we thought contributed. So those are 1,900 different ones. We saw some of them over and over, and some of them we saw only once. So of all those variants, nearly 40% of them had never been seen before. Despite doing all this, hemophilia was well characterized in a lot of ways compared to other inherited disorders when MLOF started. So it was a surprise, I think, to me for sure, as we were going through the project, that we continued to find throughout the project variants that had never been seen before that we thought were important. So this also implies that there are almost certainly many other DNA variants that we have yet to see that can cause hemophilia, and illustrates the importance of continuing genotyping efforts. Next slide. The other thing is that we saw all kinds of different kinds of DNA changes. There might just be one nucleotide change. There might be a whole bunch of the genomic region that's impacted. And so this cartoon, which if you want to go into detail, the main take home is that particularly in more severe, very low level or undetectable activity levels, which is severe hemophilia, there are some kinds of gene changes that really you need to be in a specialty lab to find them. You're not going to see them in 23andMe if you're participating in these sort of recreational ancestry or other clinical things. They need to be done in labs that are able to see all this kinds of variation. And those are labs that are expert in hemophilia. So it was great to be able to genotype all of these people together in one lab, in one method, to be able to find these comprehensively. Next slide. Another thing we were able to do, thanks to the data that was entered in the Athens data set, was we were able to, for the number of participants where there was inhibitor information, we were able to look at factors that may contribute to inhibitor risk. And so one of those, which there had been suggestions of in the literature before, but there had been some conflicting evidence, was whether or not race and/or ethnicity contribute to inhibitor risk. And so because of the large number of people participating in this single study altogether, where we have a comprehensive genotype, we were able to do an analysis. And we used the participants' self-reported identity with regards to race and with regards to ethnicity, and we were able to analyze them separately. And so we found that if someone self-identified as Black, they had a much higher rate of inhibitors than if someone who self-identified as white. And similarly, if they self-identified as Hispanic, they had a statistically higher rate than if they identified to be not Hispanic. And this was true not only in hemophilia A, but also in hemophilia B. And so this really, we hope, lays the groundwork for people to begin to investigate what is underlying inhibitor risk that differ between different racial and ethnic groups. Next slide. So where do we need to go? Picking up from Glenn's theme that he started at the beginning. Next slide. Well, thanks to the 80% of people who participated and consented to research, we now have the biggest resource in hemophilia ever created so far. And we really want to fulfill that original MLOF vision to support the scientific breakthroughs of tomorrow, hopefully today now and also tomorrow. So this My Life, Our Future research repository, it has all the genotyping information from the original genotyping effort for all the people who agreed to research. It also has biological samples, so everybody has a little bit of DNA, and we were able to use this for some additional sequencing we'll talk about in the next slide. About a third of people we were able to get a useful RNA sample from, and then about 80% of people have a plasma or serum sample, and that's a huge biological resource that will be available, that is available to the research community to continue to ask important questions relevant to hemophilia. And then, of course, Athen has been collecting valuable phenotypic data, which includes the demographic data, but also relevant clinical data, and those linked data are available to research repository people who have applied and are approved. Next slide. So MLOF also benefited greatly from the NIH, the NHLBI, the National Heart, Lung, and Blood Institute, absolutely saw the value in a large cohort of this bleeding disorder, and MLOF was accepted into TopMed. So TopMed is supported by NIH, and it is intended to support scientific advances through what's called omics. So this is big data research. Going back to what Glenn and Mark were saying at the beginning, this was one of the big goals from MLOF from the beginning, was can we get big data going and can we do big data research? And thanks to NIH, we were able to develop some big data. So we were able to get whole genome sequencing on nearly everyone in the repository. So that's DNA sequencing of nearly all of the DNA instructions in the nucleus. And then also over 3,500 participants have what's called RNA, which was that second step in Glenn's slide. You know, the DNA becomes RNA, becomes protein. And we have additional sequencing approved from NIH to do more samples that we're waiting on the pipeline for. So this is an amazing and deep resource that's available. We wanted to care to guard the privacy of everyone who participated. And so as part of that, there's a lot of application processes to make sure that only qualified scientists are accessing this. And so the process is that you can get limited access through application to NIH, and you can get access to the entirety of the repository data by applying to My Life, Our Future. Next slide. So what kinds of questions could this repository help address? We can ask questions about how factor VIII and factor IX work together in the clotting cascade. We can start looking at what kind of genotypes increase the risk from inhibitor development. We already found some, and they're in the 2022 paper. We found some new findings. How can we better define the genes that impact inhibitor risk or response? What other kinds of gene variants? These are those modifying variants that Glenn and Mark were talking about earlier, that beyond those that cause hemophilia, how are they impacting the health of the people affected? And then really, like, can we really deeply characterize and identify what is the nature of the variant, so that maybe someday soon we can be editing or fixing or replacing these genes effectively for more durable and potentially curative treatments. Next slide. So these are our contacts. Barb and I are fortunate. I feel fortunate to have been invited to be a lead investigator with Barb. You know, Barb really made the genotyping program work, and it was a privilege to train under you, Barb, in the lab during My Life, Our Future. So please feel free to reach out to us, and there will be more coming soon on the future of the repository. Certainly stay tuned. Next slide. Really, the acknowledgement list is always inadequate when an entire country comes together to make something happen, which is what happened here. So many, many thanks and acknowledgement to everyone who participated and all the people and families affected by hemophilia in the United States, and to the tremendous effort of the hemophilia treatment centers, that the existence of the network and the cooperation of the network made this work. And then, of course, many people who did the outreach, education, genotyping, coordinate the research, and just make the project work. So thank you so much. And the next slide. And with that, we want to thank you, the participants in the webinar, and we're going to invite questions, and hopefully we have a lively discussion.
Thank you very much, Jill, and Mike, and Glenn, and Shelley, and Mark, and Barb. I'd also like to welcome Dr. Len Valentino, the past CEO and president of the NBDF, who is stepping in for Mike, who had to step away. I would like to remind everybody that we will look forward to answering your questions now. I mean, this is a really exciting presentation, really remarkable research. It's really exciting to see everything that has been accomplished, and then to think about all of the things that that are still possible with this amazing collection of data. If you have any questions, please put them in the Q&A button at the bottom of your Zoom panel. You should see there, and we'll try and answer all of your questions. We'll start with, maybe I can start with a really basic question. I don't see Jill at the moment. I think she might have had to pop away. Maybe I can
No,
ask
I'm here.
Barb. Oh,
If you
you're there.
can,
Okay.
yeah.
You've vanished to me somehow.
Mhm.
Oh, you're hiding under the questions. Sorry. Let me move the question. So I wanted to ask a super basic question to start with. We talk about this as a repository, but what exactly does that mean? Does this mean everyone's DNA is kept somewhere, or is this more of a virtual repository, like a database, or like, what does that mean?
Yeah, great question. What is a repository? Well, for My Life, Our Future, what it is everyone who consented to research, we saved every bit of the blood, of the pieces of the blood sample that we could, because we not only know what we want to research now, but it's hard to predict what's going to happen in the future. So we want to keep those samples so that we can ask cutting-edge questions continually over a long period of time. So what came in, everybody got a genotype, which is a DNA test. So that meant that whatever was left over from that could come into the repository. So there are some leftover DNA samples. We decided that the NIH opportunity was a really huge opportunity for our project. Many people can do research in the data without having to go back to the sample. So what we did is we did everything we could to make sure that essentially everyone would have a sample that was able to get a very high quality whole genome sequence. And that's what we accomplished. So for some people, that was the end of the DNA from between the genotyping and the whole genome sequencing. But most people, we have a little bit left. So we're reserving that for questions where the whole genome sequencing might not be the kind of data where we would want to ask that scientific question. And we've already had a few examples of that. And then we also have, if you participated in research, if you were in My Life or Future, you might recall there were a couple extra blood tubes drawn. So in those cases, we saved them for the RNA, which we've done sequencing with NIH. And then we have plasma and sample that are kept in a freezer in the biorepository in our building. And those we keep with lots of special software and protections so that we can pull them and send them to researchers who are qualified to do the research and can ask questions to hopefully turn them into lots of more other important data. So it's a combination. It's both samples that are sitting waiting to become data, and then there's the data that we already have. Barb, did you have anything you wanted to add to how the repository is?
No, I think that was a good explanation. And I think it's important as a reminder that everyone is a code. So even though the DNA and RNA and plasma is there, it is coded. So we don't know the identity of individuals. We just go by the code.
That's a really good point, Barb. And occasionally we're asked about this, and even if I wanted to, I can't figure it out. I can't figure out who anybody is. So the links are at the Hemophilia Treatment Center, and the research is separate from the genotyping program.
There's an interesting question we received from an audience member here, and I think there's a couple of different ways we could try to answer it. So I want to kind of bring it to you for your consideration. They're asking if there's a paper or place where they can look up their mutation.
Yes.
So I think there's maybe two ways I'd like to address this. One is if someone participated. I know, Shelley, you mentioned receiving a variant report. So I'm wondering, is that something that did everybody get? Is it really clear to them how to use that? If they've lost it, could they get a new one? And then also, if you didn't participate or if you don't have such information, is there somewhere that you could look up mutations and see what they typically mean?
Maybe I'll start that. So we did not send reports to individuals. We sent to their HTCs and to many individuals at the HTCs sometimes, anyone whose name was on the an original form. And so then the local hemophilia treatment center would then put the name of the person or identify the report with the name of the person and then return that to the individual. So if someone is looking for a report they didn't receive, they should contact the HTC where the sample was collected.
Yep. So, and the second part of that was like, where can we find out about information about variants? So if you look online to the JTH paper that we published, we wanted to really make what the findings were accessible. So in the supplements is a list of the variants that were found. There's also some online databases that have more linked information. And so one is the EAHAD Factor 8 and Factor 9 databases, and anyone can access those online and see what's known about those variants. And we do have plans to try and publish or make accessible more publicly, like what do we think of the interpretation of significance of those variants in the near future. So just as another sort of giving back to the people that participated in My Life or Future. For any one person who wants to understand the significance of their genotype, it should be, as Barb said, the HTC, that facilitators, or who their provider is now, because people might have changed. Genetic counselors or providers experienced in returning genetic results are the best source of understanding what someone's personal genotype means for them, because that's always the best way to do it.
Great, thank you. There's a question here for Shelley. So there's the audience member starts by thanking you all very much for sharing the wonderful webinar, addressing not only a scientific scene, but also the approaches to living with hemophilia A and B. And the question to Shelley was about the screening of your niece and whether there's been improvement in the care that you've received in the past, or as you see maybe younger people being diagnosed now, not necessarily your niece, but you know, have you seen improvements? Have you seen changes in the way that this is being managed?
Thank you. And my niece actually wasn't impacted, which was great news for her and my family. So we were able to discover that. I think the question about how this has impacted other women accessing care is a very large question that really warrants a webinar in and of itself. I think that we are definitely moving in the right direction, and we are a lot better off than we were years ago when we really didn't understand that women could manifest symptoms and bleeding at all. So we're heading in the right direction. However, I still think it's a journey that we're on in terms of helping people fully understand the implications of women with bleeding. And I actually think Jill may be able to address this even more with a lot of the research that she's done on females and bleeding.
I can comment broadly. So firstly, Shelley, that's very nice of you. I think it is a topic where we need more evidence to make things better. So, and I do think the My Life or Future Research Repository is a great place to start laying the groundwork for many of the asking these questions in many thoughtful ways. So one of them is we don't fully understand why levels are so variable, or we don't understand why levels don't correlate with bleeding a lot of the time. And so it's going to be a group effort to answer this question. So I think the My Life or Future Research Repository is a great place to start that. And and when I say it's a group effort, I mean, it's also a partnership, I hope, going forward with people who are living with and affected by these disorders, that we do the research together.
Great, thank you. So another question that we've had, and it kind of speaks again to this question of, you know, how does this data translate into actual care, and how does it impact people's lives? And so we've got an audience member who's asking whether if we believe that the people who identify as Black and Hispanic, or if the data suggests that they are at a higher risk of inhibitors, is there then are they followed more closely? Is the larger community aware of this? Is that an important step that needs to happen? And I think, Len, maybe you wanted to speak to that.
No, actually, I was going to invite Jill or Barb to talk about that, because I believe there's a specific research study that was done in the My Life, Our Future program that addressed this particular question.
Okay, so we'll bounce to Barb, maybe.
In terms of impact on clinical care, I'm in terms of understanding it, there was a study done with a research repository in terms of how we look at it for clinical care. There is a question in the community whether there is a type of factor that, whether the type of factor may impact early inhibitor formation, though in that study, people of African origin were not really sitting a part of that study. So I think in terms of knowledge of inhibitors and knowledge of careful following at the Hemophilia Treatment Center with factor exposure, consideration now that we have alternatives to factor, whether that's something, in the balance of what's the right product for the patient, that should be taken into consideration. And, another risk factor inhibitor formation, which is independent of the genotype, but clearly it has a genetic impact. And this is why it's important to study modifiers for inhibitor risk, is that a family history of inhibitors is also a risk factor independent of the genotype. And so when we have a family who has high-risk genotype, and they have a lot of individuals in the family, then we have to be very, that really needs to impact the decision-making as to what's the right treatment for that individual. I don't know if you want to add to that, Jill.
Yeah, I'd love to take it. That was a wonderful answer. And it got me thinking as you were talking. So two things was on the clinical side. I think, and I'm hoping we're about to see the results of this research published soon. There should be soon, and there was a description of this somewhat in the original paper, that not all inhibitors happen in people with severe hemophilia, that there are some that happen in non-severe, and some genotypes are associated with that. And so for those participants, I think particularly making sure there's surveillance for inhibitor formation and discussion of alternative therapies, or minimizing factor VIII exposures if there's another option. And I think that's a place where, just in non-severe hemophilia A and B, like to have that conversation. And then of course our hemophilia B patients, the options are different for non-factor therapies than they are in hemophilia A. And I think we just need to keep learning more who's at risk. But great point, Barb, about the family history informing. It really supports that there's genetic stuff. And there's two research groups that have looked at the HLA genes, the genes that are part of how our immune system presents, like, for example, factor VIII to be a danger signal or not, and shown that there are some signals there. So that also shows that this research repository should be really helpful to look at, like, what are the factors underlying inhibitors.
And we've had a similar sort of a question, just looking at all of those variants. Is there anything that we are learning in terms of what might be critical in shaping the phenotype and bleeding? Kind of looking at something different to development of inhibitors, but are there any genotypic variants that are telling us something about that kind of relationship between the phenotype and bleeding and the genetics?
Do you want to take that one, Jill? I think that's a really important question to address.
It is. It's felt like I was talking all the time.
Yeah. And so that is an obvious question that to try to address through the MLOF Future. Sometimes with the data we have, characterizing the phenotype fully has been a bit of a challenge. And so we may need to look at getting more phenotypic data, which means like how people are bleeding, and really defining well who bleeds a lot, who doesn't bleed a lot. There may be an opportunity in the future to improve those data to help us answer that question.
Maybe a related question. You've shown that there's really a huge number of variants, and also mentioned that there probably are many more that we've not managed to study yet. Does this present a challenge then when we talk about gene editing? is it going to make it really kind of complex and very individual? I don't know, Glenn, if you want to speak a bit to that. You were talking about the potential, if I understood correctly, of some fairly tailored gene therapy, and whether that's something that's, you know, within
Yeah.
the realm of imaginable in the near future, or is that something that's still technologically quite far away?
Well, with thousands of gene variants, gene editing does become a problem, and it would be difficult to do. But there are a couple of different variants that would be amenable to gene editing. One is the inversion mutations that people have heard about, where a good part of the factor VIII gene is reversed. And so because of that variant, it can't make protein. And so that is amenable to gene editing to either unreverse it or otherwise fix that gene so that it would then be able to make protein. And that affects 40 to 45% of the total number of patients with factor VIII deficiency, severe factor VIII deficiency. And then the other one are these nonsense variants or stop codons, where because of a change in a nucleotide, instead of the gene being read all the way through, it stops at a certain point, and so you just get a partial piece of the factor VIII or factor IX. Actually, we're referring primarily to factor VIII here. And by doing that, then you don't have a functional protein. And so there's technology there are a variety of technologies actually that have been around for 15 plus years to try to read through those still called stop codons. And there are some newer, and I don't know that those worked very well in the past. They've tried to use them for muscular dystrophy with minimal success. But there's newer technologies to read through those that appear to be coming forward, and that affects about 10 to 15% of patients with severe factor VIII deficiency. So those are two examples where you can begin to reach into subpopulations and not necessarily have to try to fix everybody with the thousands of gene variants that we've got. For that, we're really still talking about putting a new gene in. A replacement gene in and just ignoring the patient's gene.
Right. I'm going to just bounce the conversation back to the inhibitor question because this is something that our audience is really interested in. And we have somebody asking about...
We can't hear you, Fiona. I can see you're talking, even though it doesn't look like you're muted.
Is this any better? Nope.
I can hear her.
I can hear
I
you.
can hear her.
Okay. Sorry, Jill.
Nope, still for me.
I'm afraid it might be just you, Jill. I'm sorry. I'm going to direct this question to somebody else.
Oh, sorry. Mine is fixed. It was my computer. I apologize.
All right, perfect. It was very selectively not allowing me to speak to you, unfortunately.
No, it's my computer.
So if you'd like to answer this question, maybe. So we're looking at this question again of inhibitors, and the audience member is asking for the people who may be at risk of developing an inhibitor, but who do not have a severe hemophilia, whether we could see by their genetics or their family history that they have a high risk of developing inhibitors despite not having severe hemophilia, whether this would be an argument to consider giving them prophylactic therapy where they might not otherwise qualify for it.
So I think I'm going to reframe it a little to be the question I think we're asking. So like what I think what we mean about prophylactic therapy, like in non-severe, that's usually driven by the bleeding. So if somebody needs to control bleeding that's unacceptable, then I would say that's a time to start thinking.
Oh, hang on, Jill, you're muted now.
Jill, you went to mute.
I don't know why. I'm
Your
sorry, my
computer didn't-
computer's rebelling. Or can you hear me now?
Yep,
Yes.
we're good.
Okay, it thinks I'm muted. Anyway, so as long as you tell me. I think prophylaxis is largely based on prevention of bleeding. So usually in a person who's not severe, we're talking about controlling bleeding that was unacceptable. But I think we might be thinking instead about a factor exposure And so you know, it is sometimes between the provider and the participant, if there's great concern that there's going to be an inhibitor, and I guess you want to know, it would be something to consider as long as it is also part of their treatment regimen that makes sense. So I think in a person who's not bleeding right now and hasn't received a lot of factor, then I think that again, between the patient and their provider, then that would be a discussion that I think where it's a little more of a gray zone, because it usually takes multiple times of the immune system seeing factor before an inhibitor happens. So if we're not anywhere near to the danger zone, they might still have a very safe treatment plan, even if they are at high risk for inhibitors. So I think this has got to be one of those very individualized conversations. But what I think is the crux of the question, I absolutely agree with, is we've got to really consider what is the immune system seeing every time we treat bleeding, and what's the risk for inhibitor every time we do that.
Yeah.
Could I just elaborate just to?
Yeah, great.
Because from a very pragmatic standpoint of, you know, someone who faces the day-to-day insurance reimbursement challenges, getting access to treatment, it's not inconceivable that we would reach a point where the genetic data coupled with the phenotypic experience that Jill was talking about could actually support our advocacy arguments and to support our access to therapy, where a genetic report with what we know is typical for someone with this genotype could supplement a request for treatment and to help us overcome either what might be viewed by a payer as a more high expensive or perhaps not a first-line treatment or some other option. So you know, I don't think we're yet ready to say this is the list of people that have to get prophylaxis, but I see this information, you know, coming together in time as we learn more with the clinical and the and phenotypic and the genotypic data coming together. So I think it supports us sort of in our individualized access as well as accessing the right treatment.
Mark, that is such a great point, and I can see Barb nodding too. So I want to say it already helps. It already helps to have the genotype. And one thing it helps with tremendously, not only in picking treatment, is to pay for the laboratory surveillance for the inhibitor in the first place. Like, why are you surveying this person who's got a factor level of 11%? Because the genotype tells me I need to look for that inhibitor because they've had an exposure. So anyway, Barb, did you have?
Yeah, no, I agree with that. It helps in a number of circumstances, and I only see that increasing. As Shelley nicely pointed out, you know, what we know about like half-life and how that would impact treatment options. So we can use that.
Thank you. And I will just ask maybe Barb, if you can just add a little clarification, because our questioner has come back just asking if it's a question of if you're going to have factor exposure in the earlier years, you're more likely to develop an inhibitor, or is it just a question of time, or what's the relationship there?
The relationship, it's mostly a lot of exposure. So when we see that, most frequently is after a procedure where an individual has received a lot of factor exposure. I might add, for individuals with mild hemophilia A, in these individuals at high risk of inhibitor formation, we can often use desmopressin and tranexamic acid and avoid factor exposure if they're not someone who needs a continual factor replacement, and then we could look at alternatives. So there are many approaches that individuals can discuss with their provider.
Right. Thank you very much. Unfortunately, that's all the time we have. I know there are a lot more questions that we could ask, and I know you have a lot more exciting things that you could share with us, but unfortunately, we're limited by the time. So I just wanted to thank everybody who has submitted a question, and I would also really like to thank Glenn, Barb, Mark, Shelley, Jill, Mike, and Lynn for sharing your time and your expertise with us this afternoon. And I really want to thank everybody who's taken the time to attend this webinar as well. The recording of the webinar will be on the NBDF website shortly after this ends. Also on that website, under the events tab, you can find all of the past Wednesday webinar recordings, as well as a schedule of upcoming webinars. So thank you so much for joining us this afternoon. Have an excellent week.